Okay. So we will have two main projects basically. So you have 6 groups, right? All of you, there are 6 groups. So you have 6 groups. So the first 3 groups will do the same project. The other 3 groups will do the same project, but each within their own group, okay, or not and reported as one group. Uh, so you cannot collaborate between different groups except just for exchanging maybe some information, small information. And this will do project 1 and the other one will do project 2. So what are the project now? So the 2 projects are basically similar, although different for different case. Yeah, the first project is related to the control of PCR chamber. Yeah, thermal control of PCR chamber. I believe all of you have known about PCR, yeah, polymerase chain reaction. Anyone has not ever heard about PCR? Anyone? So all of you have known about PCR, yeah? What does it stand for?
Polymerase chain reaction.
Polymerase chain reaction. So PCR is now basically one of the main diagnostic tool, yeah, to diagnose COVID-19, yeah, to detect the presence of SARS-CoV-2 virus, the virus that caused COVID-19 disease. So what does it do? It consists of 3 steps. The first one is denaturation, and step number 2 is called as annealing. Step number 3 is called as extension. So in denaturation, this is done at 95 degrees Celsius. So a sample which originally contained double-strand DNA, yeah, I hope you already learned this in biology with Pak Maruli, will be split, denatured into 2 single strand, yeah, with complementary sequences like this. Okay. Now, this is the step of denaturation. Then at annealing, this is usually done at, I think, 65 degrees, something like that, if I'm not mistaken. In annealing, a primer is attached to this single strand DNA. Yeah, this is double strand DNA. This is a single strand DNA. A primer means that a single strand DNA that has certain sequence that will attach to the single strand DNA and then later being extended to fill in the complementary sequence until the end primer. So the primer represents the probe of the PCR that is used to detect specific gene within that genome, within that genomic DNA, that can indicate whether that DNA is a particular species or not. For example, in the case of SARS-CoV-2 virus, usually the primer will be related with the S gene, the gene that is encoding the spike protein, or otherwise in also with N gene, some with the nucleocapsid protein, and many others. So what will happen is within that long, let's say this is long single-strand DNA from the viral genome, the prime will attach here. Okay, the primer usually is not that long. Yeah. Then another N primer will also be there. So this is the complementary DNA. Then this is another primer here. So then during the extension step, which is usually done at a little bit higher temperature, let's say 72 degrees Celsius, this in that liquid pool, there will be many uh, materials of the DNA, yeah, the adenosine, the guanine, cytosine, and thymine, IGTC of the nucleic acid base. So that will form during the extension. This will bind to the complementary sequence of this single-strand DNA to complete this whole sequence. So it becomes then another double-strand DNA for that particular gene. So yeah, for that particular, the particular genes will be amplified. For example, until here, stopping gene. Similarly, the bottom part here also. Only if we have beginning gene and beginning primer and end primer. So after that, this will repeat again. This will repeat again to cycle number 1, denaturation. So this resulted double-strand DNA along with the other maybe remaining dsDNA, which double-strand DNA which had not been denatured at the first cycle will be now denatured again. So to split becomes single-strand DNA, and then anneal again with the primer at 65 degree, then extension. So forth, yeah, until this particular gene has been amplified uh, large enough that it can be read using other instrument. In the case of COVID-19 diagnostic, so what they usually use is quantitative RT-PCR. The RT here stands for reverse transcriptase. Why is it reverse transcriptase? Because we know that SARS-CoV-2 virus, that the virus that cause COVID-19 is basically an RNA virus, an RNA virus. So the PCR cannot amplify RNA directly. So the RNA is first reverse transcripted into DNA, and then the DNA is then being amplified with PCR. That's why it is called RT-PCR. The Q here stands for quantitative. Yeah, quantitative. So why is it called quantitative? Because one of the primer, either the beginning or the end primer, is conjugated with fluorescence probe. Yeah, with fluorescence probe, for example. Let's say here. Yeah, for example, here it is conjugated with fluorescence probe. So when this particular gene is being amplified more and more, yeah, the fluorescence. So fluorescence means that it will be bright. It will be. It can emit certain wavelength upon the excitation by another wavelength. So when the amount of that DNA of that particular gene is amplified, increases, then we will have a response. If this is fluorescence, this is with time or cycle time, how many times it is cycled, then it will increase rapidly like this. So we call this this is the fluorescent intensity. Okay. So depending on whether you have the increase here or here at the red one, or maybe at this part, it will determine whether you have a lot of viral load in your body or not. So if the CT, the cycle threshold is still small, and you already have an increased amount, a lot of increased amount of the gene, it means that you are at the beginning of infection, or you are at the peak of your infection. Yeah, the viral load in your body is a lot. But when then and we get recovered, the CT will becomes larger and larger. So it means, what does it mean? It means it takes more cycle of PCR in order to amplify the particular gene that indicate, that identify the virus. Okay, until this explanation, is it clear enough or not?
Clear, sir.
Clear, yeah? Okay. So I'm not asking you for this first task to make the whole PCR device. No. Only what you need to do is to develop a system, a small chamber. Yeah. You don't need to make a small chamber. There is a small chamber. This will be equipped with heater. Okay, with heater. The heater usually can be from nickel chrome wire. Okay. Then there will be also thermistor, temperature resistor. Thermistor. Okay. Temperature dependent resistor or RTD, resistive temperature dependent. Thermistor is a sensor, a type of sensor that can indicate the temperature of the chamber as a resistance change. Okay, so then you need to convert that resistance change into voltage and put into the Arduino microprocessor to control the heater. So even you don't need to put to make the chamber later. The chamber will be made by 4th semester student. So please coordinate with them. With in this case is with Ezra and Angeli for this PCR. Yeah? And the 3 groups of you will do the control of thermistor and heater in order that you can cycle these 3 different temperature, 95, 65, 72. And as a sample, to simulate the sample, we will just use plain water, okay, small amount of water. Can you then heat it up until 95 degree, maintain it for some duration of time, then lower the temperature to 65 degree, maintain it for some duration of time, then increase again to 72 to 72 up to 75 degree, maintain it for some duration of time. After that, going back again to 95 degree, then 65 and 72, all this in cycle. Okay, so no need to put actual DNA yet, yeah, because the emphasis here is on the electrical circuit and programming the control system, because the course is electrical engineering too. Okay, while the semester 4 is in the sensor and measurement course, so their focus will be on the development of the chamber and the sensor as well as the heater. So it's better that you could also, the 3 groups that is doing this task can coordinate with that group in the 4th semester student that do that makes the chamber, so that then we can collaborate. So this one is Ezra and Angeli from the 4th semester that doing this. Yeah, so what you need to do is, for example, you have a set of temperature, for example 95. You have to have the heater to heat it up, the system. This is the system, the chamber. Then from here, it is being measured by the thermistor. This is the temperature output. This is thermistor. So here delta R, delta R representing this T out. The T out. So there will be error here. The error will, okay, so sorry, here there's supposed to be microprocessor, the microcontroller, the Arduino. The microcontroller will then send the correct signal to adjust the current or voltage to the heater, such that then it will heat up or slower or cool down. You could add additional actuator, for example, here Peltier cooling. Here Peltier cooling. So as the actuators, there could be, you can use only one heater, but then your cooling will only then depend on the surrounding air. So you may not be able to achieve rapid cooling, for example, from 95 degrees to 65 degrees. If you want to have rapid cooling, you need to control the second actuator, which is Peltier cooling component. These are all available in even Tokopedia. Okay. Any questions regarding this task number 1? Hello, clear?
Until now, no questions, sir.
Okay, no question, yeah.
Perhaps later.
Okay, so
If
I will clear here. I will draw the second task. So the second task is what I call as active temperature sensing. So the idea is to have a thermometer which is based on thermistor. Thermistor, but either we have the sensor here in our lab using carbon nanotube coated thread, but since your subject here is electrical engineering, you can also use normal simple thermistor. But so this is used just, for example, just to measure the temperature, human body temperature by touching the human body temperature using this component. Only now we have different way of measuring the temperature. Instead of using passive detect sensor like thermistor here, what you will have is also a kind of heating element as well. Okay, heater, of which then you will flow a current with a current source to this heater. So let's say this have an initial resistance of R0, right? Then you are measuring the voltage here, yeah, V output plus minus. Yeah, V output will be a function of IS and RO, right? V output will be equals to IS times the initial resistance. Now, with thermistor, okay, so this is when the temperature attached to this thermistor is changing. Okay, for example, when it is touched by a person's hand, or whatever, yeah, then so here it has, for example, initial temperature T0, then this T0 becomes, let's say, T1. So delta T, the temperature change, equals to T1 minus T0. As a result, with a thermistor, usually the resistance will also change as a function of this temperature change. Delta R is a function of delta T, temperature change. So you will have here R1 minus R0 as a function of F delta T. Some thermistor is linear, has linear relationship. Some other are not linear. So when the resistance change, okay, so now the V output will not be IS times RO anymore, but it will be IS times R1, right? The V output will be IS times R1. Then, as consequence of this value change, it means that there will be also delta V equals to V minus V out. The current output voltage across this thermistor minus the initial output voltage. Now, the idea is that what we call as active temperature sensing is using null mechanism. So this value delta V has to be made 0 again. In order that you make it 0, 0 again. So it means that you have to add minus width something like that. Yeah? So this becomes Windows, then yes, VO dash has to be add with something to make it to be equals to V0. Okay, so in that case, okay, so you have to put, wait a moment, erase first. Mathematics is a bit complex. Sometimes I'm not. So you have to put additional current that will make additional voltage. So this becomes VO dash plus, let's say, delta V increase, yeah, minus V output. So this is supposed to be 0 now. So the VO dash plus delta V increase should be equal to V output, the initial resistance, the initial output voltage. Okay, and this means to add additional delta V in, you have to add current, right? So the current now, you have to control the current. You have to control the current. So to become IS dash equal to IS plus delta IS. So that this IS dash, yeah, multiplied to R1, yeah, equals to now back to V output. Only the initial output. Yeah, so from here, then delta IS equals to IS dash minus the initial current. This will be a function of the temperature, because the temperature or delta T, the temperature is related with resistance change. The resistance change will be causing voltage change here. And to compensate that voltage change, you have to increase or decrease the current here. Yeah. So such that then this becomes 0 again, right? It means then you know the temperature not by converting the temperature directly using thermistor formula, but rather from a feedback control mechanism. Yeah, by the amount of the current that you need to inject more or you need to inject less towards this heater system in order that the output voltage remain the same. Or you can do also vice versa, that you are measuring the current, the input is the voltage, depending on which you prefer. Okay, so this is what I call as active temperature sensing. So in this sense, hopefully the sensitivity of the measurement will be higher compared to if you just use passive thermistor. Passive thermistor means that, yeah, you have thermistor, which is dependent on temperature, like this, and passively, so you just measuring the resistance here. But you don't adjust, you don't control the current flowing through that resistor. Okay, but with this, you are controlling the current amount to that resistor and adjusting it such that then the output here, the output remains the same. The output remains the same. The amount of the adjustment will indicate increase or decrease of the temperature, because always the resistance will depends on the temperature for any conductor usually. So this is for second task. So I will put this in general to the Google Classroom, and between the 6 group, Please later comment which group would like to do task 1, which group would like to do task 2. Okay. Yeah. First things first. First come, first serve. So please, whoever gives comment first, then we'll have the right. Yeah. Both have its own difficulties and easiness as well. Okay, not necessarily this second task is more difficult than the first one. So the first one can be also more difficult than this first one, because at the first one you are using 2 different components for the heater and for the thermistor. But in this case, you are using a single heater. You are controlling the current. Based on that control of current, you can deduce the temperature increase or temperature decrease. Any questions so far?
Sir, one group make one or make 2.
Make one report at least and one demonstration.
Or one
Although,
demonstration and one report.
yeah,
Okay.
in one group choose between this task 1 or task 2. But if one of the tasks, for example, let's say task 1 already chosen by 3, then no choice. The remaining group should do task 2, the project number 2. Any questions?
Sir, when do we
Sir.
choose it? Oh,
Who first? Jesse or Catherine? Jesse, yes.
okay. When do we choose, sir?
Yeah, as soon as I uploaded it and you put it as comment, similar like some
Oh,
time ago, right? When for your in the electrical engineering lab, you choose also the topic. Yeah.
okay.
So this project is for the the marking of electrical engineering as well as electrical engineering 2 laboratory. So this will be the semester project for the lab as well. Okay. So later the evaluator will be myself and Kak Devita, as well as maybe I will ask Pak Aulia to join.
Okay, sir.
So there will be milestones. Okay. So maybe week 10, I'm thinking of putting this assignment not in the electrical engineering classroom, Google Classroom, but instead in the electrical engineering lab, Google Classroom. Yeah. So we will have milestone as well. So maybe in addition to the normal module, we'll see later. Yeah, it could be in the electrical engineering lab session, or it could be in this electrical engineering 2 session. Okay, and I hope we can have at least 3 milestones before the final presentation. The first milestone, at least by tomorrow, not tomorrow, sorry, next week. Each group will present shortly, 5 minutes. So 5 times 6 will be 30 minutes maximum, about what they understand about the project. Yeah, from reading, from what, and maybe some initial planning. Then milestone 2, you should already have had some basic circuitry or schematics of the circuit that you want to implement, as well as immediately you should also order some remaining components that you need. Kak Devita may have sent you already the basic component, the Arduino, multimeter, those things. But some additional component like the sensor, the heater, we may need to purchase again. So please immediately, if you could tell Kak Devita as early as possible, then she can purchase it for you and send it to your home. Now, yeah, one group, one demonstration, one report. However, since we are now in school from home, so and the members of the group may reside in different cities, even so we don't expect you to meet with the other members of the group. Okay, just do it and communicate and do it together, maybe using Zoom session or Google Meet session, whatever. Do it together so that then you can correct each other. Any questions?